Hello everyone, how are you doing? So I want to have an overview for yesterday's lesson, and also I'm going to cover some general information for today's lesson in order to get rid of some misconception and if any part of the yesterday's lesson was not understandable for you. Okay, so yesterday we talked about RNA mRNA making, which we call transcription. So we previously learned about the replication, which we're making copies of DNA from the template DNA strand. Now we're talking about the transcription, when DNA will be transcribed to RNA. Well, again, it's the same language, so it should not be that much problem, not that much differences. Basically, they use the same rule. That's the common field between the replication and transcription. Base pairing of the nucleotides matters. So let me see what stuff I can cover to get rid of your misunderstanding. Okay, so I'm gonna draw a double-stranded DNA. As you can see, just like I put the space between two, and imagine there's a gene here, part of DNA that I want to transcribe it. I want to make mRNA, messenger RNA, from this part of DNA, this gene. Alright? So, remember that in biology, in genetics, do you remember from replication that the DNA polymerase used to add the nucleotides to the 3' end of the previous nucleotide, so it cannot be to the 5' end. Let me show you something from previous information. If it's a base here, sugar here, here you have another carbon and phosphate groups with this, and here this is number 5, carbon number 5, and here is 3, okay, and OH is here. This is the nucleotide. So I used to put the head here to make it funny for oxygen. I used to maximize the oxygen here. So sugar, deoxyribose for DNA, and 3' end and 5' end phosphate. So you have to add another phosphate and another group to here, basically to make the chain. So everything is added to 3' end. All right, does it make sense? So let's get back to that, those two strands, because we're adding to the 3' end. The direction of what we're making is going to be from 5' to 3'. Doesn't matter if we're making the copy of the if you're copying the DNA, the new strand is going to be made from 5' to 3'. If we're making the RNA, we're transcribing again, it's going to be from 5' to 3'. But there's a difference. Basically, in DNA synthesis, DNA polymerase has to use the primer should exist, because DNA polymerase cannot add anything to the 5' end, and needs the primer by the help of primase, the enzyme that makes the primer. But in RNA polymerase, the enzyme that makes the RNA for transcription, does not have that problem, can start from the scratch. But again, direction of the newly made strand is going to be from 5' to 3' end. Okay, let's get back to the strand. So here's our gene. We want to make the mRNA from that. Okay, this is called template. This is template strand, and we make the mRNA here. All right. This strand is complementary to the template strand. All right. This is non-template. I think some of you had confusion about this part. Non-template. mRNA, let me just make some nucleotides for you. Imagine we have ACG here. ACG on the template. What we're gonna make, so here is gonna be just like CG. A will be matched to the T, but we do not have T, so we make UGC, correct? Here, remember the non-template is complementary to the original template strand. Is gonna be again TGC. If you notice, the mRNA's sequence resembles non-template DNA sequence for that part. Okay, so basically they're exactly almost the same, excepting wherever you see U in the mRNA will turn into, should be T, substituted with T in the non-template strand. Because they're the same, exceptionally, I don't know who came up with this idea, we call non-template strand as coding strand. So a lot of you had confusion here. Basically, intuitively, you may say, okay, the original one, the template one, which codes for mRNA and the protein, should be coding, but not really. Basically, coding strand is the strand, the complementary strand of the DNA that the sequence matches the mRNA. That's the trick. All right, I hopefully made it clear to you. All right, so let me see what else I can say. Okay, now we can go to the details for today's lesson. The piece of DNA, the strand that specific gene was supposed to be transcribed. Let me make it bigger. Imagine from here to here is the gene that we're going to transcribe. All right, so what is the enzyme responsible for? What is the enzyme responsible for transcription? RNA polymerase, right? This enzyme is just one universal one for prokaryotic cells, prokaryotic organisms, but in eukaryotic organisms you have different types of the RNA polymerase, which mainly the one that is responsible for making mRNA is basically RNA polymerase 2. So there is this part from here to here, from a start point. So here on the chromosome, on the DNA, there's a part here. You can see that. There's a part that's called promoter. This is the sequence of nucleotides that RNA polymerase will recognize it and will attach it. Specifically, there is a nucleotide here. This nucleotide is the start point. It's inside the promoter, okay? And also before, by the way, it's very important to know that here, this area, if I make it, let me put the direction for that. Imagine this is from 5' end to 3', or let's make it just like different direction because we want our I mean, it doesn't matter, but 3', 5', or whatever direction you consider. So here is the upstream, like a river, upstream to that area. This area toward this area. If it goes this direction, if you go in this direction, this area is downstream. So upstream upstream to a start point, there's a few nucleotides that we call it TATA box, TATA. So here's the TATA box, TATA, that enzymes, we're talking about the eukaryotic RNA polymerization, transcription. So this box, this sequence here before or upstream to start point will be recognized by some enzyme that an RNA polymerase will dock into that site, near that site. These are the factors that the RNA polymerase will recognize and start the transcription. There are enzymes in prokaryotic cells, prokaryotic organisms. RNA polymerase does not have any problem, recognizes start point very easily. But in eukaryotic cells, there are a lot of enzymes and protein factors are involved. We call them transcription factors. Without them, RNA polymerase cannot attach to those areas. Without a specific transcription factor, one of them, a TATA box will not be recognized, and RNA polymerase cannot dock into that site. Is that clear? Hopefully. So, when RNA polymerase attaches to the site, it will open two strands. Well, I had to make it a little bit of space, but basically they are very close together. Now RNA polymerase, when docks onto this side, the promoter side, it will open two strands. And if you remember in DNA replication, there was an enzyme to do that. It was helicase, basically, to do this responsibility. But in yeah, it was the helicase, but the RNA polymerase itself opens two strands from each other. It doesn't need any help from the enzyme. Helicase was for replication. Remember helicase, replication? You know helix? If you don't remember that the helicase is responsible to pry it open, remember the helix, double helix. Helicase opens the two strands from each other. So when the two strands are open for, this time, transcription, starts this side for transcribing and elongating. So initiation has started, now elongating. The same rule for the same rule as replication happens here for transcription. While we gotta take into account that in replication you need primer. In transcription you do not need primer. So RNA polymerase is capable of doing the transcription. It's a lot of terms. Transcription of the DNA to make the RNA without any primer from the scratch. Basically, this is the way they say that. And yeah, it continues until there should be a termination part, right? Again, in prokaryotic, remember those, let me erase that to explain. In prokaryotic, you had UAA codon, UAG, and UGA. Whenever the RNA polymerase sees these, equivalent nucleotide on the DNA, which is not going to be UAA, it's going to be ATT and others. If you see these termination codon, for example in mRNA or DNA, it's not going to make any amino acid. That's the end of it, and the RNA polymerase will separate. But in eukaryotic cells, it's a little bit different. So it's not like this. There's a specific sequence, and at the end of the, could be everywhere, towards the downstream of the transcription that happens. If I'm not mistaken, it's the sequence of AAA, UUAAA. Let me check to make sure I'm not mistaken. They call it polyethylation signal. So it gives a signal to RNA polymerase. That's the end of it. But RNA polymerase goes a little bit further, for example 40 or 50 nucleotides away, and then separates from the strand, and you have the end of it, the transcription. Okay, that's the termination part. So in prokaryotic, UAA, UAG, and UGA is the end of it, but in eukaryotic you need that polyadenylation, that sequence, to be recognized by RNA polymerase, and a little bit later it's separation of the RNA polymerase and the strand. That's the termination. So in prokaryotic, that's the end of it. The mRNA is ready to do everything, right? To go for translation. Sometimes transcription happens and translation of the other, on the other hand, happens simultaneously, right? But in eukaryotic, it's not like that. In eukaryotic, what you're making is pre-mRNA. To have a mature ones, you have to do some editing and to add some stuff to modify the pre-mRNA to make it mature RNA, mature mRNA. So imagine this is the pre-mRNA. This is the pre-mRNA, okay? When the mRNA is synthesized a little bit for 40 or 50 nucleotides, something will be added while it's in nucleus. Something will be added. It's a guanine, special guanine with the 3 phosphates added to the 5' end here, to the 5' end of the pre-mRNA. And when the transcription is almost finished, is finished, at the 3' end, something will be added by the name of poly-A tail. Poly-A tail. A lot of adenine will be added to this side. Okay, so these two will be added. This one at the beginning, almost at the beginning, and this one at the end. But they happen in the nucleus. Remember, in the nucleus. Adding these two will help the 5' end cap, we call it cap, and poly-A tail will help the mRNA to pass through the nuclear pore and gets inside the cytoplasm. Plus, the cap and poly-A tail prevents newly made RNA to be degraded by some nucleases inside the cytoplasm. That's very important. So two main, and also these two parts will help, especially the cap will help the mRNA to dock onto ribosome for translation. So 3 main goals: protection, what else I said, recognition by the ribosome, and also the last one I myself forgot. The last one was, you tell me. I can't hear you, but you tell me. So protection. Oh, also preventing passing through the nuclear pore and protection from the nucleases. All right. So here, when this is added, there is another modification. Remember, a gene has different parts. You have the parts that should be expressed for that part of gene. They're called exons. And also you have the parts that you do not need to express them. They're not going to be translated. The red part, the exon, will be expressed, but intron will not be expressed. Introns will not be expressed, okay? So, introns will be removed by the help of something is called spliceosomes, which is the mixture of the RNA, the ribozyme. It's kind of specific RNA. It's like enzymes. Remember, we used to say enzymes are protein, but not all of them. This is the exception. Specific RNA inside the spliceosome will cut the introns from the 5' and make them loop and get rid of them, and then attaches the exons together. All exons will be attached together, and this is the one. I mean, it's gonna have the 5' cap and 3' poly A tail, 5' cap, and plus there's something that here and here we call it here is the 3' untranslated area, UTA. It's not going to be translated this, but it's going to be in the mature mRNA. Here you have the 5', this piece, 5' UTA. Can you see it? Sorry, it's a UTR. UTR. U stands for it's not going to be untranslated, if I'm not mistaken. So these parts are not going to be translated, but will help just like for translation. Introns will not be in the mature mRNA, but UTRs will be in the mRNA. Okay. So you have this whole exons connect together. These are the part of both exon and intron are part of the gene, but exons express exon execution express. You can justify to yourself to remember it. It's gonna be in the final mRNA. This is the mature one. It's gonna go to ribosome and be translated by ribosome and other enzymatic things that. So remember we talked about, I will put the video, we talked about the spliceosome. That's the complex that just like cuts the intron and takes it out and connects the exons together. What else we can say? If from here, this concept, the cutting the introns out, we call it the splicing. What do we call it? Splicing. Okay? Splicing is part of the modification that happens. Adding 5' cap and poly A tail is part of modification. A splicing could happen in specific ways too. Let me show you this picture. That causes different proteins are made. If I just like choose exon 1 and 2 to be in the final mature, it's gonna make a specific protein. If I choose 3 of them, all 3 of them, for example, if I have A, B, C, I have A, B, C. What if I just have A and B in the final mRNA, or I have 3 of them together? It's going to be different type of protein. That's why we have, for example, around 30,000 genes, but we can make a lot of proteins, much more than that number. It's because of the alternative splicing. Different, exons can match to get to attach to each other. So what do we call this? Alternative RNA splicing. All right. Also remember that there's another thing that's called shuffling, or mRNA shuffling, if I'm not mistaken. What does your book call? Specifically, your book calls it exon shuffling. That it's about 2 different genes this time. So the gene here with intron and exon, for example, crossing over can cause some shuffling that happens between the exons of 2 chromosomes. Exons of 2 genes of different chromosomes. So I try to make yesterday's and today's lesson clear for you. Remember, the exons are the ones who are expressed, and splicing causes the introns to be degraded, cut off and degraded. And what else I can say? Every single You know, the proteins have domains. Domains in protein, for example, an enzyme have catalytic domain or binding domain, different domains. For example, A in this case may be catalytic domain with functional group. B would be just like active area. I'm just making it up. And the C could be just like specific area, a specific domain to attach to part of the organelle. Just put it that way. Hopefully I made the stuff clear to you, and yeah, good luck with tomorrow's. By the way, be present in office hour today, because I'm going to talk about the pedigree. I'm going to talk about the pedigree, and it's very important. All right, take care, guys.